You will have to accept my thanks for the invitation. I have been most often reminded, being from utanför riket i Sverige, men någon person som aldrig gjorde det var Viktor Mutt och Tamas Bartfai. Och det mest underbara samarbeten var med dem. So in English, I am extremely lucky that I have been able to work with Tamas Bartfai, who took me to Viktor Mutt. And Viktor Mutt looked at me behind that enormous piles of papers and said to me, I like Tamas, who was sitting in front of me. I only could see Tamas, because he makes beautiful pictures and he is not rushing with telling me what they mean. He is telling me what he finds, where he finds them. And I thought that this picture, which I have been in possession of because Tamas Bartfai gave it to me, it has many interesting people on it. First, in the first row, and this picture has so many people who have been making major contributions to neuroscience. Eccles. Here we have Tamas Bartfai, you just heard. I don't think I have to make a plea for that he has made his share. Pernov, who worked for von Euler and has been an extremely important neuroscientist at the Karolinska. Floyd Bloom, editor-in-chief of Science, I became the successor of, and who said, if you were good enough for Bartfai and Mutt, I'm sure you should be my successor. And trust me, there were many people who wanted to get the department, which was moving between Salk and Scripps. And it was only the question of where you want to be. I was asked, where do you want to be? And I said, well, I don't know. I have to ask the Karolinska, because they appointed me to Bengt Samuelsson's chair. And the Karolinska said, being Kansviksa, you can choose any one of them, they are fine. And on the one condition, you don't resign at the Karolinska. And I said, why? He said, nobody ever resigns from the Berzelius chair. So, and as you see, somebody could lose a hair in peptide research. Here is Jean-Pierre Joncheux, Jan Lundberg, whom you have heard of, Costa, Menek Goldstein, who has been not only a wonderful mentor to Fuchse, Högfeldt, and many others, but who has done an incredible work, very modestly always. The only person as modest as Viktor is Menek. I really will, unlike Tamas, from a perspective of a biochemist, talk about why it was so remarkable and remarkably successful, the work of Viktor Mutt. Here you see a man who worked on an extremely commonly occurring neurotransmitter. Nobody believed him. They ridiculed him first. It's Bernstock, purinergic transmission. Do you mean that ATP is a transmitter now all of a sudden? This was the first question before he entered the room fully. So, but he survived. This is Marian Schulzberg, Lars Tarenius. So, Eric Kandel. So as you see, this is a picture important to have if you ever have a publication. Maybe Tamas has the copyright of it. He made it, or he has had it made. But I am very fond of it. This is the only picture which I took from every every place I worked. First it came up on the wall at Yale in the room of Ritchie. And Ritchie is fundamental to neuroscience because he discovered the voltage-sensitive sodium channel. And without that, there is no excitability. And when I came there, he said, "Did you bring some luggage?" I said, "Yeah, I brought my 3-month-old daughter, my clogs, and this picture." So he said, "I would like to have it." So under a while, it was hanging at Yale in Ritchie's room. At that time, Kendall was no Nobel laureate, and Freud was very, very important at St. Elizabeth. But in his room, Viktor was the most important because he produced as a very modest, incredibly diligent, and not ever commanding, but always advising colleague. Now, as a biochemist, you will understand that the main issue is that if you want to do something which had to do, and you have to remember, with purification and sequencing, and you have heard about Jörnvall. Jörnvall has sequenced almost everything what Viktor Mutt has purified to homogeneity together with Europae. And before you think that he hasn't been involved with industry, Vitrum, which was responsible for insulin production for Sweden, in which was later incorporated to Kabi Vitrum, was a company of Europae and Mutt. So he did understand it. He just was not interested in companies and money. He lived in his rental apartment with Birgitta, which I often visited, and was by far the most modest, significant scientist I knew. I did not know a nicer, more modest man. But as a biochemist, you will understand that the main issue is that if you want to identify and purify and sequence things, then you need a lot of starting material. This has put him into a very complicated place. The only two people who have used such enormous amount of starting material to isolate a peptide were Andrew Shiley and Roche Guillermin, who have isolated the hypophyse releasing factors. They each used 1.5 million hypophyses of sheep. Now, it's important that Viktor used porcine. Using sheep or porcine, species whom we humans consume, permitted that if you isolated something, there was no committee on Earth who would prevent you from injecting it to humans and look for is there any biological effect or any pharmacological effect. It was incredibly much smarter than isolate the same thing from anglerfish or pufferfish, which would kill a human. You isolated it from porcine, probably millions of humans have been exposed to it already. It was incredibly smart. Nobody at that time thought about it. And why I think that this is so important for us is to remember Viktor, because he was an extremely clever biochemist who has worked on a set of peptides which at the beginning were only considered, if anything, as hormones. But the known peptide hormones immediately brought into the imagination and the knowledge of all physiologists insulin. And that's very big compared to the peptides, which Viktor worked with. The second problem was that, as we all learned, insulin is made as a prohormone, and there are several enzymes needed to cut and to remove the C-peptide. So it is fair to say that while somatostatin was known through the work of Roger Guillemin, who was on Saturdays 100 years. As you see, we are watching a generation here, a generation of giants. And when I said that there are roughly 1,000 drug targets, I should also say that when I entered Roche as vice president, there was a sign which my predecessor left, and it said, safe drugs find many applications. So if of the 1,000 drug targets, 50 will be well drugged, that will mean 200 indications in which it will be used. This is how it will be. So the significance of this is far beyond what you can see today. And maybe Tiit and Ulo got a little discouraged by that not yet there is a drug on galanin, but galanin was a fantastically good project to train on, because you were one of the first ones to do an L-ala mapping of pharmacophores of a longer peptide. It has become a strategy which is used today everywhere. They do it now, not by synthesis, but by mutation. But that comes from your work, Tiit. If you take all those publications that are in the thousands, they do it with mutations to find the pharmacophores, but it's the same, or to find the important residue in a receptor, whether to connect to a G protein, to oligomerize, or to bind a ligand. And therefore, I would like to say that I will jump over many slides, so that don't worry, that the most important things which Viktor, as a good biochemist, has understood. Can you see if I'm standing here? No, you can't see. Why don't you say something? The most important thing was, which I, as a poor physicist, when I came to biochemistry in Stockholm, was told that you have to have a very sensitive assay, and you have an enormous amount of starting material. And he fulfilled this. Nobody ever talks about it, because they thought that it's natural. It was not natural at all. In fact, there was not a single person at the Karolinska Institute, including his boss, who didn't make fun of him, that if you want to smell where the Karolinska Institute is, you will find it because Viktor Mutt is cooking tons of porcine gut. And that is a smelly project. But they didn't understand that if you are looking for something which is about 1 ppm of the total amount, then you need a lot. And the second thing, you need a very sensitive assay. And in the beginning, he has been working on insulin release from pancreatic islets, which was then by far the most sensitive bioassay you could think of. Not only was it extremely important for public health, since insulin had to be made and calibrated for the diabetics of Sweden, and so later on through Novo and Nordisk insulin for the world, but and first, which also used the same assays. The second thing, which I think was the most important by far, was number 2. The concept that while there are hundreds and hundreds of peptide fragments, proteins are either disappearing in the proteasome after ubiquitination, or they are chopped off by exo and endopeptidases. And if you find a protein which is not taken by a C-terminal endopeptidase, which are very active enzymes, then it is most likely having some signal function. And this was an insight which has led his work. So the practical and very, very important 2 things, large amount of material, and large I mean tons, and sensitive assay, I mean more sensitive than femtomole per liter is detected, which was then nothing else than hormonal assays. And then the concept that we should look for C-terminal. And they very clearly, it was not a mistake. This is as you read. Chemical determination of polypeptide hormones, fragmentation analysis, and carboxy-terminal amidated groups. That's what they were looking for. It was not an accident. And they first made two-dimensional GLs and chromatograms of C-terminated amides of every amino acids. And then they were looking peptide like this. And after that, they had a very well worked out strategy. They made antibodies, polyclonals at that time, later monoclonals, to peptide fragments, which they were using not to measure the peptide. There were no radioimmunoassays or ELISAs around yet. Yarlow and Peter Perman did not publish them. But they used it as an immunoadsorbent to identify other family members of the peptides. It was incredibly smart, because then you all of a sudden could use the same extracts. Not only did do that, but also showed you that long before you could clone the cDNA and find out peptides which are through processing of it and translation of the processed mRNA would give you glucagon and glucagon-like peptide and GLP-2 or GIP. And this is how they discovered families. And they also tried to purify the receptors. They have asked us to synthesize peptides which they biotinylated. At that time, there was no more high-affinity binding than biotin-avidin binding, and they have tried to isolate receptors this way, and sometimes they were successful. And they followed the literature. When cDNA techniques became available, they have used every peptide which they knew the sequence of, and there is a whole group of people we don't talk about, but the former president of the Swedish Academy, Dan Larhammar, made his whole career on cloning based on the neuropeptide Y sequence, all NPY cDNAs, NPY receptor cDNAs. And thereafter, the peptide had to be also studied for its distribution, and that was happening by synthesizing the peptide and labeling it. The most common labelings were 125 iodines, either by chloramine T or by Bolton Hunter. And this was thanks to that Ulo Langel has been diligently and rapidly synthesizing the peptides, and we had the best of possible collaborations with, first of all, with Bruce Merrifield at Rockefeller, who invented solid-phase peptide synthesis. We had Anders Sundén, who did the PhD. You guys have gone to pretty good PhD places. Didn't you find an iron response element somewhere at the National Cancer Institute? I think you have gone to, all of you have gone to very good postdoc places. I believe that if they go to better postdoc places than they were doing their PhD, that can only be good. And the most important is, when we come to drug targets, that it turns out that these receptors are all seem to belong to the GPCR class B. And this means that these have many binding sites, not only for the peptide, but also for negative and positive allosteric modulators, and they often oligomerize. And you may say, so who cares? But in fact, the first most well-sold drug, the 1,4-benzodiazepine, also known Valium, is just an allosteric modulator of the GABA ligated chloride channel. Of course, we didn't know it because it was discovered for its anxiolytic effects by Leo Sternbach through a phenotypic assay in anxious mouse and in anxious wife of his own. At that time, you could do clinical trials at an N of 1. But I think that this is my main slide. I will jump over many, because I rather prefer that you follow the genius involved in these seemingly trivialities. Find something which, if you're successful, can be put into humans without any kind of ethical issues and any kind of problems of it will be antigenic. Porcine definitely is such. Find a very sensitive assay. At that time, 1,000,000 times more sensitive than any other. Find an ID which distinguishes the peptide you look for. And sometimes you're lucky. So the porcine galanin is C-terminally amidated. He had been looking for human galanin, it's not amidated. But it has 80% sequence homology. I will not use 40 minutes. So I give a few examples of the papers, but the papers are only interesting because before all this standardization and age factors, these papers were landmarks. And you see, nobody is anymore talking about data sleep-inducing peptide, but they understood that the antibody can be used to define motifs. And this was after a short discussion with the monoclonal, with Köhler coming over, and I brought him to Viktor. And since the monoclonal antibodies was made at the Roche Institute of Immunology at that time in Basel, the very first time, we were rather interested in what is the size of the epitope of an antibody. And Viktor immediately said, but that could be a motif in a peptide family. So even if it did not become a very important family, they understood the methods. And then, very, very rapidly, they were on to that there may be the same hormone be processed in different cell types differently. This is the case for the glucagon gene, processed differently. This is the case for GIP. And what is, people were always saying, and Viktor himself was onto it, to find out which are the most important glucagon fragments. Now, Tamas has overemphasized that we would, in my opinion, that we would like to have antagonists to some of the peptide receptors. But in fact, the most sold and most, today, most important, not only commercially, but endocrinologically, clinically, neuropeptide, gut peptide ligands are the GLP-1 receptor agonists. And the most sold ones are actually peptides. Semaglutide is a peptide modified in positions 2 and 8 and given a big fatty acid chain, which increases not only its half-life from 2 minutes to about 5 weeks until it's 0. Unfortunately, it also gets it into the brain. So brain receptors are mean. And so, and not to be forgotten that peptides have a very, very short half-life. So if you synthesize GLP-1, for example, it has a half-life in humans, only 2 minutes, because usually the number 2 amino acid is a key pharmacophore. Tiit was the first one to show that in galanin, the key pharmacophore was tryptophan number 2. The same dipeptidase cleaves it. Once it takes it off the first two amino acids, there is zero effect at any of the three colonin receptors. If you take only one, then it still works at GALR3. And so Merck, which is not liking to make peptides, made a good inhibitor. That's Januvia, DPP4. So this you have seen, and I won't go into much, other than that substance P research was not such a tremendous disappointment, because when we talk about pain medicines, we are all looking for pain medicines for three kinds. One, which works in inflammatory pain. And to be honest, ibuprofen is very hard to beat. Cerebrex was to beat it, but it had a major flaw. It was only tested in 15,000 people, which was considered an enormous clinical trial. But once it had no side effect in these, it went into 5 million people, and 1,248 of them died in heart attack, which they didn't have any indication of before. So Merck, one of the largest and best companies, almost went under, since the juries in the US have deemed such enormous fines. And finally, it had to be a class action suit. And it was not the fault of Merck, and it was the fault to imagine that when you have a safe drug, that you will be able to control how many people take it. But of course, no drug is safe. So when we are looking for a pain drug, we look for inflammatory pain. We look for pain which is functioning in bone and cancer pain. And we all know that morphine is not that great in it. And therefore, we are looking for everything. So therefore, we put the NK1 antagonist, which was a big disappointment in sensory pain, into people with cancer pain. And those people are usually suffering also from nausea and emesis because of the chemotherapy. And there is today absolutely no better drug than the NK1 antagonist. And this is coming of substance P research. I would argue that the number of cancer patients who is helped by this is enormous. Enormous. So last year, I looked it up, last year there were 420 million doses sold of it. So I don't think it's a failure. And it goes into sick people, really sick people. They are already clinically diagnosed and treated for their cancer. And they are treated with chemotherapy because we don't have anything better and more targeted. So I don't think it's a big failure. I think it's a pretty good thing. And this is showing if the drug itself is safe, it will find an indication. The other one is a little bit more speculative, but it does work. And if you will talk to anybody who has experienced hot flushes, you will say that that's not a small potato. Okay. So here is a list of which you have seen from Tamas, the peptide receptors, and I added colonin because the people from whom I took the slide did not have it. And I have taken one because this has been something made me very, very angry in the last weeks. As you might have followed, for the GLP-1 receptor agonists and their use in obesity and in type 2 diabetes, Hoffmann has received the Breakthrough Prize. I think it's a very well-deserved prize. What I didn't like is that when they asked me to support it, they asked me to put in a sentence about GIP. The peptide Viktor discovered simultaneously, And the sentence would have said basically that GIP was kind of a mistake of a genius a genius of Viktor, because he didn't understand that it's not GIP, it's GIP1 which is important. But if you look at it, you can read the second line. GIP and GIP1 were discovered at the same time. But if you read the second line, trizapid is the first dual agonist. It has been just approved. And just read the text. The two together is by far better than anyone alone. Was it? And it's a real fixed dose combination because they are covalently linked, 1 to 1. Can it be better? Can it be more clearly better stoichiometry? So I think even on this, Viktor was not beaten, not that he ever considered it as a competition. He was happy when he discovered something new, and he hoped that it will be interesting. He never used the word to be useful. He said curiosity. So the other thing which Tamas has brought me into was the coexistence. These peptides, which he has discovered, obviously from porcine gut, I will be how many minutes do you want? Okay. The peptides he discovered were from the gut. The question is, are these peptide or peptide families occurring in real neurons? Can you isolate them from the brain? And one of the first ones isolated from the brain or from nerves was the one neuropeptide Y here. It's a novel brain peptide, and it was isolated from the brain. And we worked on it. The lab has worked on it. In fact, we produced the very first paper on the NPY receptors, and the ligand we used was iodinated NPY, produced by Ulo Langel synthetically and then iodinated, and then Anders Sundén has shown its binding, and we also showed this was the very first indication that these receptors are G protein coupled. I have gotten from Lefkowitz a very big lecture on, then one doesn't shoot away all these big findings in 2 papers instead of writing 50 on them. Maybe this is why I am not going to join them together with Kobielka. But what is important is that these collaborations, which came from the gut peptides, translated into the peptides, which are also isolated from the brain. And as you see, the distributions, which I all take from different papers, from Tamas, from Schafuchse, and from many others, show that they have a tremendous possibility. And as I mentioned, not only NPY agonists and antagonists, but the subtype specificity, since there are 5 subtypes. And since the peptide itself is 36 amino acid long, and it has 3 pharmacophores, give you the idea that there may be many drugs coming up. So galanin you have heard an awful lot about, and I hope by now Ulo has forgiven me the enormous amounts of times he had to synthesize galanin and galanin with different alanines in the positions. This is the one which he and Tiit have studied, and this was the first peptide in which absolutely systematically everything was scanned with L-alanine replacing any amino acid to find out whether it's a pharmacophore or it's part of a pharmacophore, that amino acid. This is now standard technique. Okay? So, I don't want to go much on it. I only want to say that the number of papers, if it were nothing else than to train brilliant students to become better, then Galanin would have been the best topic to work on. Because what did we do? Not only we determined the pharmacophores, we determined the active fragment, it would have taken for us, not being a drug company and not having a big library and not having expressed the receptor as it is today done, very long time to develop antagonists and agonists. ULO has undertaken to synthesize many agonists and antagonists which were peptides. So for the periphery, they were intrathecally applied, and for the CNS, they were applied the ICV application, mostly by Jacqueline Crowley, and later on by others. There were some small molecules synthesized around the tryptophan number 2 by famous chemists and by not so famous chemists, but all of them had a weak affinity. Now weak, I mean, is micromolar, but you should remember that acetylcholine is only binding at micromolar affinity to its receptors. Neither the nicotinic nor the muscarinic receptor was ever isolated by using acetylcholine, its endogenous ligand. They were isolated by mustard gas, and they are isolated by bungarotoxin or torpedotoxin, which bind to it. They are highly not endogenous ligands. So to make galmic and galnon to be micromolar agonists is not so bad. Not so bad, I should argue. And then there are plenty of studies on its coexistence with serotonin and acetylcholine, and with dopamine and noradrenaline. Very early we have shown that there are inhibitory effects in this one. Mena Goldstein was involved. These are papers, many of them with Fuchse. These are many with Gonzalo and Ladinsky, and it was the basis of a very large collaboration with the Mario Negri Institute. And then we did what is today people are doing. We have generated the antagonists, also as small molecules. None of them is as good as the ones which Ulo has made when he made M35. And as molecular biology became more facile, we have generated a galanin overexpressing mouse, and we were looking for some effect in the CNS. And we immediately found with our collaborators that the seizure threshold was dramatically changed. And that was a very big deal. And there are still 4 large programs ongoing, because as you know, in epilepsy, we are not doing extremely well. If you think about it, we don't have too many new epilepsy drugs. We have made GALR1 knockout mice, GALR2 knockout mice, and today people have made transient knockouts. So I will not go any longer. I can say that my gratitude to Viktor can be summarized like this. When I was elected by the US, this was the text under it. This is for me. It's not for Viktor, but it is thanks to Viktor and to Tamas. You are being honored for contribution to Neuropeptides, including muscarinic receptors, but pioneering work on neuropeptide signaling systems. The pioneering part came from Viktor and Tamas. I am standing on their shoulders, and I thank you for your time. I hope I did this.
Thank you very much. Maybe there are questions we have.
I don't think so. Is anybody?
Then I use this microphone and ask one question. I was listening very carefully both lectures. You were both talking about peptides, about neuropeptides, and so on and so on. And I expected that you say a few words about replacement of peptides with peptidomimetics. So maybe you just say that this is a very bad idea and will never work and so on, but maybe you just have some other comments.
So I have a slide which will, this is irrelevant. Who I am is totally irrelevant. The work is more relevant. So GPCRB, to which class of G proteins, these peptides which Viktor and others have discovered, have two ways of being regulated. One is, of course, the binding of the agonist, like galanin, like NPY, and they bind to an extracellular large domain, which is much, much larger than from other GPCRs like the beta receptor, beta adrenoreceptor. But they also have both negative and positive allosteric modulator sites, which have been quite well proven at the level of structure. Cryo-AEM is not expensive anymore. And so together with the AlphaFold, the people in London who sold their company now back, Google from the beginning supported them. It's now part of Alphabet Healthcare, AlphaFold. So together with them, we have looked at, there are maybe 16,000 identified allosteric sites which are on GPCRs. And this does not include the sites where a GPCR with its 7 transmembrane domain is oligomerizing. And one of the first things we did with galanin, which was also one of the first things one has done with the GPCR, that we have shown that galanin receptors form homo and heterodimers. So if you make a yellow fluorescent protein GALAR1 and a green fluorescent protein GALAR2 and express them in the same cell, you can study whether they homodimerize or heterodimerize. That was the very first study we did. Now it's trivial. Now everybody has to do it. It's like, became like in gymnastics. There is an obligatory program you have to do if you are competing in gymnastics, and then there is a special program which you choose yourself. But this is now the ELASCAN, the active fragment study, the oligomerization, the homo and hetero oligomerization. These are now part of the absolute every GPCR B goes through. And this means that we have now a very large number of sites to be drugged. And so antagonists will be used as negative or positive allosteric modulators. The people who are either have a company tradition to sell peptide hormones, like Nudisk insulin and Novo. I should say here, Nudisk insulin, the lab where Ulo was with Chimoncic, we made Nudisk insulin with yeast codon usage. So it all of a sudden the same yeast made 12 to 18 times as much insulin than if you use the human insulin nucleotide coding. Of course, it was from the best Carlsberg beer brewery yeast. This is what the Danish explanation by it is. So. But anyhow, Novo Nordisk became one company. And they like to sell peptides. So semaglutide and Wegovy and Ozempic are peptide drugs because they really feel that they are in the business of selling peptides and they have made their fortune together with Lilly in the US. Novo made its fortune with insulin, and Hus did it too in Europe with insulin. And so they are very wedded to make improvements with formulation, like the insulin pen, which one can laugh at. You say, is that a big invention? I can tell you, my mother used to go to the nurse down to get her insulin shot together with some other well-dressed ladies. It took them half an hour to get dressed for that. The time I brought home the first insulin pen, she said, I don't have the time with this anymore. So formulation with peptides is a different business compared to oral drugs. So they will be non-peptidic, mostly allosteric site targeted drugs, and there are. They are now in clinical trials in epilepsy for sure, a new narcolepsy drug, which is also non-peptidic, is in phase 2, and there are 4 antidepressant approaches based on NPY and galanin receptors, and they are all non-peptidic because they are aimed to be oral. Now, I would like to make, before you all go home and take ketamine or some magic mushroom after you listen to Tamas to say, that this is not major depression. This is what is the approval says, not the use. The approval says is for treatment-resistant depression, what ketamine was approved. Which means, treatment-resistant means that it didn't respond to the tricyclic antidepressants, which we have used 65 years, in more than 1.2 billion times. And when I say a time, means a 60-day period of treatment. It means that it not responded to the SSRIs, which we have used more than 4 billion times, 90 days at least. Because they were safe. In fact, today, SSRIs beat anxiolytics because chronic SSRI has less side effects than a benzo. I am sorry to say it after having come from Roche, but it is so. So ketamine was not approved to treat depression. It was approved for treatment-resistant depression. Now then it became a big industry around it. It's an incredibly dangerous drug. And when we are playing ketamine plus something, we are taking the edge of how dangerous it is. In most places, ketamine was thrown out because of the psychomotor side effects it gives. It is terrible. If you are part of general anesthesia and you look people at the wake-up, you can see that the nicest, kindest people coming out and waking up are saying the most horrific, profane, violent, unbelievable things. If you omit any ketamine or ketamine-like drug from their anesthesia mixture, they don't do that. It's not trivial. So I think that today the majority of antidepressants remain SSRIs because they can be given by your general practitioner, and because it's safe. And safe always beats efficacy, because efficacy can be achieved by placebo. Safety cannot. Thank you very much.